Imagine something microscopic, I mean truly tiny, something that makes one of your own cells look like a giant skyscraper. It has no engine, no power source, can't feed itself, nothing. Yet this tiny thing can somehow sneak into that skyscraper, hijack the controls, and force the whole building to just churn out thousands and thousands of copies of itself. It's this idea of a borrowed life, really, one thing completely taking over another. Okay, let's unpack this a bit. Today we're diving deep into the fascinating and, let's be honest, sometimes pretty scary world of viruses, and also their even weirder relatives, prions. We've been digging into a chapter from Campbell Biology in Focus, and our mission here is to sort of cut through the dense science, pull out the really key insights, and figure out why these incredibly small agents have such huge power.
Yeah, it's truly captivating, isn't it? How these particles, often smaller than a cell's own ribosomes, the protein factories, can just orchestrate a complete cellular takeover. It's amazing. So we'll unravel their unique structures, look at the really ingenious ways they replicate by, like you said, borrowing life from their hosts. We'll try to grasp their role as pathogens, obviously, and then finally, yeah, delve into that really puzzling world of prions. We're going to try and connect these concepts step by step, paint a picture without the textbook diagram so you can really get a handle on their impact.
Okay, so let's start with maybe the most basic question, one that biologists still kind of wrestle with. What is a virus? It feels like a paradox. They aren't cells. You can actually crystallize some of them, like salt, but they cause disease, and it really pushes at the edges of what we even mean by life. Fundamentally, a virus is an infectious particle. It's a piece of genetic material, could be DNA, could be RNA wrapped up in a protein coat. And sometimes, yeah, they have this extra layer membrane envelope, almost like a disguise. And the scale, I mean, the smallest are just unbelievably tiny, maybe 20 nanometers. Millions could fit on a pinhead. The largest, though, can get up to, what, 1,500 nanometers? Almost big enough to be seen with a basic light microscope. It blurs the lines.
Right, and that scale, that simplicity, it drives home that "are they alive?" question.
Yeah.
Most biologists land on this idea that they're in a shady area between life forms and chemicals, leading this kind of borrowed life.
Yeah.
And the why is crucial. A virus just doesn't have the machinery. It can't reproduce on its own, can't carry out metabolism outside of a host cell. They're basically, these highly specialized packages of genetic information that absolutely have to plug into a living cell's operating system to do anything. No energy production, no protein synthesis machinery. They rely entirely on the host.
And the genetic info itself, the genomes, they're incredibly diverse, which I guess speaks to how adaptable they are evolutionarily. We usually think DNA, right? Double-stranded DNA. But viruses? Oh no. Their genomes can be single-stranded DNA, double-stranded RNA, or single-stranded RNA. Just amazing variety. And it might be one single molecule, linear or circular, or sometimes it's even split into multiple pieces. But what's really striking isn't just the type, but how little information they need. The smallest might have, what, 3 or 4 genes? The largest, maybe a couple thousand. Compare that to bacteria, often thousands more. There's incredibly efficient genetic machines.
Absolutely. And that genetic material is protected by the capsid, that protein shell. Capsids are built from these protein subunits called capsomeres, often forming these really beautiful geometric shapes. Some are rod-shaped, helical, like tobacco mosaic virus. Think of a spiral staircase made of protein. Others are polyhedral, like adenoviruses, which look like a 20-sided die, and icosahedral. And then you get the really complex ones, especially the bacteriophages, the viruses that infect bacteria. Phage T4 is a classic example. It looks almost like a lunar lander with an elongated icosahedral head for the DNA and this intricate protein tail assembly that acts like a syringe to inject the DNA.
Wow.
And then there's that extra layer you mentioned, the viral envelope. Lots of animal viruses have this. Influenza as a prime example. These envelopes are fascinating because they're actually stolen partly from the host cell's own membranes. So it's host phospholipids and membrane proteins, but studded with viral proteins, glycoproteins, that are crucial for getting into the next cell.
Like wearing a coat made from your last victim.
Exactly, a borrowed coat. And some viruses even pack a few of their own enzymes inside the capsid, ready to go as soon as they get in. Everything is optimized for invasion and replication.
Which brings us right to their core strategy. Viruses replicate only inside living host cells. It's not optional, it's absolute. They're called obligate intracellular parasites. Literally, they're just packaged sets of genes in transit from one host cell to another.
Yeah.
The host cell becomes this unwilling factory, right, providing everything. The building blocks, like nucleotides, the enzymes, the ribosomes for protein synthesis, tRNAs, amino acids, even the energy, the ATP. The virus just brings the blueprint.
Total biological freeloading. And that dependence, it creates a really important limitation, the host range. If we connect this to the bigger picture, each virus can typically only infect a limited range of host species, or even just specific cell types within a species. It comes down to that handshake idea. Specific proteins on the virus surface have to bind to specific receptor molecules on the host cell surface, like a very precise lock and key.
Ah,
Some viruses have a broad range. West Nile can hit mosquitoes, birds, horses, humans, but others are super narrow. Measles only infects humans. Cold viruses usually stick to the cells lining your upper respiratory tract. HIV needs specific receptors found only on certain immune cells. Specificity is key.
so once that handshake happens, that binding, the cycle kicks off.
Yeah.
Generally, the virus gets its genetic material inside the host. How it gets in varies. Those T-even phages, the bacteria attackers, they literally inject their DNA. Other viruses might get swallowed whole by the cell endocytosis, or if they have an envelope, it can fuse directly with the host cell membrane, sort of melting in. Once inside, the viral genome takes command. Viral genes redirect the host machinery. Stop making host stuff. Start making virus stuff. Copy the viral genome. Synthesize viral proteins. Then, amazingly, the new viral nucleic acids and capsomeres spontaneously self-assemble into new virus particles, hundreds, sometimes thousands of them. And finally, they exit the cell, often rupturing it and destroying it in the process, which is what causes many disease symptoms. Oh, and a key point for RNA viruses, many use their own special enzyme, an RNA polymerase, to copy their RNA genome, because host cells don't usually have machinery for that.
Yeah, that's crucial. And what's fascinating here is seeing these strategies play out with bacteriophages. They're probably the best understood viruses. They show these two main alternative reproductive paths, the lytolytic cycle and the lysogenic cycle. The glycolytic cycle is the quick and deadly one. It ends with the host cell bursting lysis. That releases a whole flood of new phages to infect neighboring cells. Phages that only do this, like T4, are called virulent phages. The steps are pretty straightforward. Attach, inject DNA, hijack the cell to make viral parts, assemble new phages, and then bam, the cell explodes. Very effective for rapid spread. But then there's the lysogenic cycle. This is stealthier. It allows the phage genome to replicate without destroying the host cell, at least not immediately. Phages that can do both cycles are called temperate phages. Phage lambda is the classic example.
Okay, so how does that work?
Well, after the phage DNA enters the cell, it typically forms a circle. Then it integrates itself into a specific spot on the host bacterium's chromosome. Now it's called a prophage. Most of the prophage genes are switched off. The bacterium lives, divides, and every time it replicates its own DNA, it replicates the prophage DNA too. So all the daughter cells inherit the silent viral genome.
Wow, so it's hiding in plain sight.
Exactly, but it's not necessarily permanent. An environmental cue, maybe UV light, certain chemicals can trigger the prophage to cut itself back out of the chromosome and switch over to the destructive lytic cycle. And here's a really important medical point. Sometimes, while it's integrated as a prophage, a few viral genes are expressed, and these can change the host bacterium's phenotype. For instance, the bacteria that cause diphtheria, botulism, scarlet fever, they're only harmful because they carry prophage genes that code for potent toxins.
That's incredible. The virus makes the bacterium dangerous to us.
Precisely.
So, faced with this constant onslaught from phages, what can bacteria do? It must be a constant battle. It really is an evolutionary arms race. And bacteria have evolved some pretty sophisticated defenses. First, just basic natural selection. If a bacterium happens to have a mutation that changes its surface receptor so a phage can't bind anymore, it survives. Its descendants thrive. Simple defense upgrade. Second, if the phage DNA does get in, bacteria have these things called restriction enzymes. They act like molecular scissors, chopping up foreign DNA at specific recognition sites. Their own DNA is protected, usually by having metal groups added to it at those same sites. And then there's the really amazing one, the CRISPR-Cas system. We hear about CRISPR for gene editing now, but it originated as a bacterial immune system.
Yeah, it's fantastic.
Bacteria basically keep a library of DNA snippets from phages that previously infected them. These snippets are stored in their own genome in regions called CRISPRs. If that same phage, or a related one, tried to infect again, the cell uses RNA copies of those stored snippets to guide Cas proteins, another type of enzyme, to the invading phage DNA.
Sure.
The Cas proteins then cut the phage DNA, disabling it. It's like an adaptive immune system for bacteria.
It's a beautiful example of coevolution, this constant back and forth. Okay, so shifting from bacteria to animal viruses, things get a bit more complex, partly because our cells are more complex. Many animal viruses have RNA genomes, and many have those envelopes we talked about. That envelope is key for entry. Those viral glycoproteins studded on the envelope bind to specific receptors on our cells. After replication, new viruses often acquire their envelope by budding off from the host cell membrane, could be the outer plasma membrane, or even internal membranes like the nuclear envelope for herpes viruses.
And that bedding, does it kill the cell?
Often, not immediately, which is why some viral infections can be persistent. The cell keeps churning out viruses. Herpes viruses are a great example of persistence, too. They have double-stranded DNA replicate in the nucleus, but then their DNA can just hang out, dormant, latent inside nerve cells, sometimes for years. Stress can reactivate them, leading to recurrent cold sores or genital sores.
Right, the gift that keeps on giving.
Something like that. Now, focusing on RNA as the genetic material in animal viruses, there are several variations. Some RNA genomes can directly function as mRNA, translated by host ribosomes right away. Others serve as a template to make complementary RNA strands. One strand acts as mRNA, the other as a template to make more genome copies. This requires that special viral RNA polymerase we mentioned earlier. But here's where it gets really interesting. The retroviruses. HIV, the virus that causes AIDS, is the most famous example.
Okay.
Retroviruses are RNA viruses, but they carry an enzyme called reverse transcriptase. This enzyme does something incredible. It uses the RNA genome as a template to synthesize DNA. It reverses the usual flow of genetic information DNA to RNA, hence retro.
So it makes DNA from RNA.
Exactly. Once HIV enters a host cell, reverse transcriptase makes a DNA copy of the viral RNA. This viral DNA then enters the cell's nucleus and integrates into the host's chromosomal DNA. Now it's called a provirus. And unlike a prophage in bacteria, a provirus is generally a permanent resident of the host genome. The host cell's own enzymes then transcribe that proviral DNA back into RNA molecules. These RNA molecules serve two jobs. Some are mRNA for making viral proteins, and others are packaged as new viral genomes for the viruses that assemble and leave the cell.
That's deeply insidious, becomes part of your own DNA.
It is a very effective strategy for long-term infection.
So, given all this complexity and weirdness, where did viruses even come from? If they're not quite alive, it's still a major puzzle in biology. The leading hypothesis is that they probably evolved from mobile genetic elements. Think plasmids, the small DNA circles in bacteria, or transposons, jumping genes, pieces of DNA that can move around within a genome. The idea is that maybe these bits of nucleic acid somehow acquired genes for protein codes, which allow them to exit one cell and enter another. They became infectious particles. But this whole picture got really shaken up maybe 15, 20 years ago with the discovery of giant viruses. Mimivirus, for example. It infects amoebas. It's almost as big as a small bacterium. Has a huge double-stranded DNA genome with over 1,000 genes. Some genes thought only to exist in cellular life.
Yeah, those discoveries were mind-blowing.
And then Pandora virus, even bigger, more genes. We even found one, Pithovirus sabaricum, that was viable after being frozen in Siberian permafrost for 30,000 years. 1.5 micrometers long.
It really challenges our neat categories of life, doesn't it? Blurs the lines significantly.
Absolutely. But even with these questions, their close link to cellular life makes them incredibly powerful tools for molecular biology research. They help us understand fundamental processes in their hosts.
Okay, let's talk about their impact as pathogens. Obviously, this is where most people encounter viruses. In animals, how do they actually make us sick? Well, there are several ways. Viruses can directly kill the cells they infect, maybe by triggering the release of the cell's own destructive enzymes from lysosomes. Some viruses cause infected cells to produce toxins, or sometimes the viral components themselves are toxic. And the severity often depends on the tissue. Your respiratory tract lining, damaged by a cold virus, can repair itself pretty well, so you recover. But damage to nerve cells from polio, for instance, is permanent because most neurons don't divide and replace themselves.
And sometimes the symptoms aren't even the virus itself, right? It's our immune system reacting.
Exactly. Fever, aches, inflammation, a lot of that is your body's defense mechanisms kicking into high gear to fight the infection.
So prevention is key. Vaccines. How do they work again, broadly?
Broadly, a vaccine introduces your immune system to a harmless version or a piece of the virus, maybe a weakened or killed virus, or just specific viral proteins. Your immune system learns to recognize it and builds defenses, antibodies, specialized immune cells, so if you encounter the real virus later, your body is already primed to fight it off quickly.
And we've had huge successes. Smallpox eradication is the prime example. A terrible disease wiped out globally thanks to vaccination. Partly feasible because it only infected humans, no animal reservoir to hide in.
Right. And campaigns continue for polio, measles. We have effective vaccines for mumps, rubella, hepatitis A and B, and many others. They're one of public health's greatest triumphs.
But if you do get infected, what about treatments? Antibiotics are out, obviously.
Right. Antibiotics target bacterial processes, useless against viruses. Antiviral drugs are much harder to develop because viruses use our own cellular machinery. So targeting the virus without harming the host cell is tricky. But we do have antivirals. They often work by targeting enzymes that are specific to the virus. Like acyclovir for herpes inhibits the viral polymerase needed for DNA replication. Or AZT for HIV targets that unique enzyme, reverse transcriptase.
Okay.
For HIV, the approach now is often a multi-drug cocktail. Using several drugs that target different steps in the viral life cycle makes it much harder for the virus to develop resistance. And speaking of HIV resistance, there was that fascinating discovery about receptors. HIV uses CD4 as its main receptor, but it also needs a co-receptor, usually CCR5, to get into immune cells. It turns out some people have a natural genetic mutation where they lack functional CCR5, and these people are highly resistant to HIV infection. That discovery led directly to drugs like Maraviroc, which blocks HIV from binding to CCR5. Real bench-to-bedside stuff.
That's amazing. But viruses keep evolving, right? Which leads to these emerging viruses that seem to pop up out of nowhere.
Yeah, here's where that constant evolutionary dance gets really tangible for us. Emerging viruses, they suddenly become apparent or suddenly increase their geographic range or impact. HIV is a classic example. It was circulating in Africa for decades before exploding globally in the 1980s. West Nile virus suddenly appearing in North America in 99. Ebola, first identified in 76, but causing massive outbreaks more recently. Zika, chikungunya, the list goes on.
So what drives this emergence? There are a few main factors. First, mutation, especially RNA viruses. Their replication is sloppier, no proofreading like with DNA replication, so they mutate constantly. These mutations can create new strains that our immune systems don't recognize. That's why we need a new flu shot every year as the virus keeps changing its code.
Okay, mutation. What else? Second, the spread from small, isolated human populations. HIV might be an example of this. A virus exists in a remote area, relatively contained, then changes in human society, increased travel, urbanization, blood transfusions, whatever, allow it to spread rapidly to a wider population.
Makes sense.
Third, and this is a big one, spread from other animals, zoonoses. About 3/4 of new human diseases originate in animals. The animals act as a natural reservoir where the virus circulates, often harmlessly.
Like bats for coronaviruses or birds for flu?
Exactly. Influenza is a perfect case study. Influenza A viruses infect birds, pigs, horses, humans, lots of animals. Major pandemics often arise when different strains mix. Remember, the H/N spikes on flu viruses. H for hemagglutinin helps it attach. N for neuraminidase helps it release. Different strains have different H's and N's.
Right, H1N1, H5N1.
Precisely. Now, imagine a pig gets infected with the bird flu strain and a human flu strain simultaneously. Inside the pig's cells, as new viruses are being assembled, the RNA segments from the bird virus and the human virus can get mixed up, reassorted into new combinations. This genetic reassortment can create a brand new virus with, say, the pathogenicity of the bird strain, but the ability to easily infect human cells from the human strain. That's thought to be how the 2009 H1N1 pandemic strain arose. It had bits from bird, pig, and human flu viruses.
A dangerous cocktail.
A very dangerous cocktail. And that's the worry with things like avian flu, H5N1. It's highly lethal in birds, occasionally infects humans with high mortality, but doesn't transmit easily between humans yet. If it reassorts or mutates just right to become easily transmissible, that's a pandemic scenario.
Scary stuff. Any other drivers of emergence?
Yes. Fourth, just changes in the environment or human behavior. Building roads into rainforests increases contact with animals harboring unknown viruses. Deforestation does the same. Climate change is another factor. Warming temperatures can expand the range of disease vectors like mosquitoes, bringing diseases like dengue fever or Zika to new regions. It increases the odds of jumps between species.
So it's a complex mix of viral evolution and human activity.
Definitely. Now, viruses weren't just an animal problem. They hit plants hard, too. Estimates are around $15 billion a year in crop losses globally. Over 2,000 types known.
Wow. Are they similar to animal viruses?
Structurally and functionally, yes, broadly. Most plant viruses have RNA genomes, often helical or icosahedral capsids, just like many animal viruses. The main difference is how they spread. Plants have rigid cell walls, so viruses usually need help getting in. Horizontal transmission happens from an external source, maybe through damaged plant tissues caused by wind, injury, or herbivores like insects munching on leaves. Insects are major vectors. Even pruning shears can spread them. Then there's vertical transmission, where the plant inherits the virus from its parent. This can happen through asexual reproduction, like taking cuttings from an infected plant, or sometimes through infected seeds. Once inside, they can spread throughout the plant tissue via these little channels called plasmodesmata that connect adjacent plant cells. Some viral proteins actually help enlarge these channels to make movement easier.
Can we treat infected plants?
Unfortunately, not really. There are no cures for most viral plant diseases, so the focus is mostly on preventing spread controlling insect vectors, using certified virus-free seeds or stock and breeding plant varieties that are genetically resistant to common viruses.
Okay, so we've covered viruses extensively, but you mentioned something even stranger. Prions. What does this mean when we talk about something simpler than a virus but just as devastating?
Yeah, prions. Infectious proteins. This is where biology gets really, really weird. They completely challenge our basic ideas about infection and inheritance. These agents cause fatal degenerative brain diseases, things like scrapie in sheep, mad cow disease, BSE in cattle, and in humans, Creutzfeldt-Jakob disease, or Kuru, which was linked to cannibalism in New Guinea. And their characteristics are just alarming. First, they're incredibly slow-acting. The incubation period can be 10 years, 20 years, even longer before any symptoms show up. This makes it unbelievably hard to trace the source of infection and means it can spread widely before detection.
A silent spread.
Exactly. Second, they are virtually indestructible. Normal cooking doesn't kill them. Disinfectants often don't work. Even radiation might not stop them. And crucially, there is no cure. Once symptoms start, they are fatal. So this raises that fundamental, almost philosophical question. How on earth can a protein, just a folded chain of amino acids, no DNA, no RNA, be infectious? How can it replicate?
Right. It seems impossible based on everything we know about biology.
Well, the leading model, developed largely by Stanley Prusiner, who won a Nobel Prize for it, is this. The prion is actually a misfolded version of a protein that's normally found in brain cells. Everyone has the normal version. The theory is that when this misfolded prion protein bumps into a normal version of the same protein, it somehow causes the normal protein to change its shape and misfold into the prion form too.
Like a domino effect.
Yeah.
Or a bad influence.
Exactly like that. A bad apple spoiling the barrel. This misfolded prion then goes on to convert more normal proteins. They start to aggregate, forming clumps or plaques in the brain that disrupt cellular function and eventually kill neurons. That's what causes the disease.
Just a chain reaction of misfolding.
A chain reaction of misfolding. It was a radical idea, initially met with huge skepticism because it broke the mold of infectious agents needing genetic material. But the evidence now strongly supports it, and this mechanism is even being investigated as potentially playing a role in other neurodegenerative diseases like Alzheimer's and Parkinson's, which also involve protein aggregation. Still much to learn.
Wow. Okay, so today we've really journeyed through this microscopic world. We've unpacked viruses, their structures, that borrowed life replication, the olytic versus lysogenic cycles, bacterial defenses like CRISPR. We looked at animal viruses, envelopes, retroviruses like HIV, making DNA from RNA, the mystery of their origins in those giant viruses, and their huge impact as pathogens, leading to emerging diseases through mutation, animal jumps, and environmental change. And then we finished with prions, those infectious proteins, defying biological norms, causing devastating slow-burn diseases through a cascade of misfolding.
Yeah, and if we try to connect this all to the bigger picture, the fact that prions exist, infectious agents without any genes, it really forces us to question our fundamental definitions of life, of inheritance, of what can cause disease. It's profound. And the constant evolution of viruses, their emergence and adaptation, it's this powerful reminder that biology is never static. It's always in flux, always presenting new challenges and new surprises. So maybe the question to leave you with is, how does understanding these tiny, powerful agents change your perspective on disease, on evolution, maybe even on the nature of life itself?
A lot to think about there. Thank you for joining us on this deep dive into the world of viruses and prions. Until next time, keep exploring.